Thank you for watching the August edition of the AJP-Heart and Circulatory Physiology video table of contents, or from the editor's desk. This month, we have published 6 outstanding review articles, including 1 guidelines paper, 1 editorial focus article, and 17 original research papers, including 1 rapid report. This is a large issue. I know you want to see what's inside, so let's take a peek at what's inside this month's issue. Seals et al. Provide an important review that summarizes the opening keynote presentation overview of the American Physiological Society's Conference on Cardiovascular Aging, New Frontiers and Old Friends, held in Westminster, Colorado in August 2017. Here they summarize work that relates to vascular aging, including endothelial dysfunction and elasticity. They discuss lifestyle changes such as exercise and diet and alterations in oxidative stress in the aging process. In a review by Cocciolone et al., they summarize the elastin protein biochemistry, self-association behavior, cross-linking process, and multi-step elastic fiber assembly that provide large arteries with their unique mechanical properties. They present measures of passive arterial mechanics that depend on elastic fiber amounts and integrity, such as the Windkessel effect, structural and material stiffness and energy storage. They discuss pathological consequences of abnormal elastin production and function. They conclude with suggestions for future research and opportunities for therapeutic interventions in genetic and acquired elastinopathies. In the third review of this issue, Mallet and co-workers discuss the mechanisms and therapeutic potential of intermittent hypoxic conditioning. They discuss clinical and animal studies where intermittent hypoxic conditioning improved capacity and decreased arrhythmias in patients with coronary artery or pulmonary disease, and produced robust, persistent antihypertensive effects in patients with essential hypertension. The protection afforded by intermittent hypoxic conditioning develops gradually and depends on beta-adrenergic, delta-opioidergic, and reactive oxygen-nitrogen signaling pathways that use protein kinases and adaptive transcription factors. In our continuing effort to provide guidelines to improve rigor and reproducibility, Lindsay et al. Provide a perspective and recommendations for use of statistics in cardiovascular research. They discuss issues related to this journal and best practices for statistical testing, power analysis, and evaluation of outliers. This important paper should provide direction for our readers and the greater cardiovascular research community. McGregor-Das and Das reviewed data on how microRNAs translate and regulate the mitochondrial genome in normal and disease conditions. Mitochondrial dysfunction occurs in many human pathologies such as cardiovascular disease, diabetes, cancer, and neurological diseases. These studies have shed some light on regulation of the mitochondrial genome, as well as helped to explain the role of microRNA in altering mitochondrial function and the ensuing effects on cells. For this review, the focus is on the role of a subset of microRNAs known as mitomeres in mitochondrial function. The authors discuss the potential mechanisms used by these nuclear-encoded microRNAs for import into the mitochondrial compartment. Tema et al. Hypothesized that small conductance calcium-activated potassium, or SK channels, are activated during hypoxia via the calcium calmodulin-dependent protein kinase-dependent pathway. They used normotensive Wistar-Kyoto rats and spontaneously hypertensive rats as a model of cardiac hypertrophy. Hypoxia-induced abbreviation of action potential duration occurred earlier in ventricles from SHR versus WKY rats. Apamine and SK channel blocker prevented this abbreviation in SHRs in both the early and delayed phase of GH, whereas in WKY only the delayed phase was prevented. In contrast, SHRs were less sensitive to glibenclamide, an ATP-sensitive potassium channel blocker, which inhibited the action potential duration abbreviation in both phases of GH in WKY rats. They conclude that activation of SK channels leads to the action potential duration abbreviation and sustained ventricular arrhythmias during simulated hypoxia, especially in hypertrophied hearts. This mechanism may result from phosphorylated calcium calmodulin kinase 2 bound SK2 channels. Finally, Ramos et al. Examined the relationship between circulating microRNA expression and variable dose exercise exposure. They hypothesized that certain circulating microRNAs respond to changes in exercise intensity and duration in a dose-dependent fashion. Muscle release of circulating microRNAs may then deplete intracellular stores, thus facilitating gene reprogramming and exercise adaptation. Graded exercise in humans evoked circulating microRNA responses that followed three profiles: non-responsive, miR21 and miR210; responsive to exercise at some threshold without dose dependence, miR24 and miR146A; and responsive to exercise with dose dependence to increasing intensity, miR1, or duration, miR133A and miR222. In mice, low-intensity or high-intensity treadmill running showed that in high but not low-intensity training, plasma microRNA 133A along with decreased intracellular microRNA 133A and increased serum response factor, a known miR133 target gene in muscle. These data support the notion that they directly mediate physiological adaptation to exercise, potentially through depletion of intracellular stores of muscle-specific microRNAs. Thank you for watching this month's installment of From the Editor's Desk. We hope you'll take a look at all the content on our website. We look forward to your comments and feedback as always, and please send us your best cardiovascular research now.